Okay, good.
Well, I guess we can say good morning, good afternoon, and good evening to all on our call. So thank you everybody for being here. And obviously want to thank Dr. Bjornsson for taking on our official first research project, and appreciate his relationship with the organization before it was even founded. So I think we're at officially 5 years since our first introduction to Dr. Bjornsson, and want to thank him for taking his time out of his day. I know that we usually meet at the ASHG conference, which is obviously virtual this year. So we're getting a glimpse before he enters that meeting tomorrow. I think, correct, Dr. Bjornsson?
Yeah. Absolutely. Thank you so much for having me, and thanks for, I'm going to try to make the most with the funds that you guys have provided. So
wonderful. Well, without further ado here, we'll let you get started.
So I'm hoping you guys can see my slides. Yeah.
Yes.
Okay. So
yes, all there. Thanks.
I got 2 tasks for today, and it's always a little harder because, like Libby said, the meeting hasn't happened, but I was going to mention a couple of things which I think is major progress, and then I wanted to give an update of where we are with the funded project. It's been a little bit slower than expected for various reasons, but I still think we're making good progress, and I kind of wanted to do both with this lecture. So just a reminder, I talk about this every time I talk to your group, but just want everyone to be on the same page. So just a couple of slides in the beginning. I kind of think about the epigenetic machinery as the genome's highlighting system. And, it's a way for cells to know which part of their genome they should be using, because all the cells have exactly the same genome, and yet you only use a small portion of it in any given cell type. And one of the ways you achieve that is through epigenetics. And what these are modifications of DNA or the associated proteins. It's not the DNA sequence itself, and they're heritable through cell division. And these marks are somewhat reversible and dynamic, and they're known to be affected by the environment. So it's a little bit exciting to think about the ability to be able to manipulate these marks for therapeutic purposes. And if nothing else, these marks have the potential of adding to the information content of DNA. And I use a very strict definition. I usually only talk about 2 categories: DNA methylation, which I'm not going to talk about today, and then the histone tail modifications, which is what we'll be talking about today. And so the DNA is not floating around in cells in kind of a linear formation. It's very highly packaged. And the simplest unit of that packaging is the histone octamer, which has these tails that stick out. So the DNA is wrapped around the octamer, and then the tails kind of stick out of that. And these tails can be modified, and the modifications kind of are seen either in open or in closed chromatin. For instance, these modifications here, H3K4, which is just the 4th lysine of H3, and these triangles, which are the histone acetylation marks, they're only seen in open chromatin. However, there are other marks like H3K9 or K27 that are seen in closed chromatin. But I'm going to focus on H3K4 because that's where Wiedemann-Steiner and Kabuki, that's the site that the factors that are mutated in those disorders plays a role. And we now know of many of the disorders, and I'm just going to focus on 3 disorders for this introduction: Wiedemann-Steiner Syndrome, Kabuki syndrome, and then Rubinstein-Taybi. You see, they all affect these kind of open chromatin marks. So they're kind of on the top part of the schematic. And The two factors that affect Kabuki syndrome and Wiedemann-Steiner are called MLL2 or KMT2D, or MLL1 or KMT2A. And Rubinstein-Taybi has two factors, Krebs binding protein and EP300. And then there's another cause for Kabuki called KDM6A. So I just want to show you where these lie and what they lead to. And the other thing that I wanted to show in this schematic is that the domains between KMT2D and KMT2A are pretty similar. They both have, the yellow mark here is the set domain. This is the enzyme function domain of the protein, and they both catalyze adding histone marks to this location. And then they have many other shared domains. You can kind of see the little colors here, how similar they are. There's a blue domain, there's a pink domain, there's these green domains. It's not exactly the same, but they're all there. So, and why is this important? So Wiedemann-Steiner syndrome, I think of this, and I think of this as a disorder of histone methylation, and it's dominant. So, it's enough to have one mutation in this gene to get, you don't have to hit both alleles to get the problem. And kids and adults with Wiedemann-Steiner have variable intellectual disability. They have postnatal growth retardation and constipation. And the frequency we always talk about is 1 in 100,000, but it's probably more common. Just based on the Facebook group and so forth, I think it's more common. And so again, what this does is it places methyl marks in this location of the histone tail. And this mark is only seen in open chromatin, and you never see it in closed chromatin. And these are just a couple of figures from Wendy's paper. And then I just wanted to show you, this is the average figure for Kabuki syndrome. And there are some similarities. I kind of feel like when I meet these kids and adults in clinic, the Kabukis remind me of the Wiedemann-Steiners and vice versa. And that's kind of how I got drawn into this early on. I want to tell you that there's a paper finally coming from some of the data that we've collected. As you know, we had been collaborating with Wendy and hoping that she'd get her monster paper out. And I still am enthusiastic about that and hopeful that that will happen. But there's also another group at CHOP, which has been collecting data from multiple sites. And I think that paper will come out in 2021. And that's going to be very helpful for clinicians because we have all these kind of little papers, and there's 1 or 2 sizable papers, but having additional big papers is very helpful to kind of go through the list of things, what you can expect. And I think that's going to help clinicians all over the place provide better care. So I'm enthusiastic about that. And I just wanted to let you know that that's in the works. There is some overlap between Kabuki and Wiedemann-Steiner, but there's also some overlap between Wiedemann-Steiner and Rubinstein-Taybi. And this is a paper that came out just this year, 2020, where they're talking about this. They've spent the whole paper just kind of discussing this thing, that Wiedemann-Steiner and Rubinstein-Taybi have similar phenotypic overlap. And one of the things that was always so striking to me, I kind of felt like Wiedemann-Steiner looked like Kabuki a little bit, but the constipation issue is very strong in Rubinstein-Taybi. So, it's kind of curious to me that this keeps coming up. Not only have I noticed the overlap, but other people have as well. And this paper came out just this year. Just a mention about Rubinstein-Taybi and why I think you should care. So this is a disorder of histone acetylation, not histone methylation. Also dominant, but there are 2 different genes, Krebs binding protein or EP300, that can lead to Rubinstein-Taybi. And the kids have intellectual disability, some dysmorphic features. This is the typical feature, which we don't see in the other disorders, that the thumb kind of points a little bit backwards and kind of classical Rubinstein-Taybi, but they have all kinds of other things. And the frequency is thought to be 1 in 100,000. And again, it's this problem with open chromatin. There's a defect in the writers that put on these open chromatin mark called histone acetylation, so that you probably can't open chromatin as well as someone that doesn't have such a mutation. And the reason why I started on my journey towards Kabuki and Wiedemann-Steiner is really these kind of relatively old papers now. This is 16 years old, where a group took mice with Rubinstein-Taybi and they treated them with HDAC inhibitor, something that inhibits the enzyme that removes this. And there were two different groups that did this, Alarcon et al. And Wood et al. And they did it in a very crude manner. They just injected the drug into the brain. So it's not very translational in nature. It was more of a proof of principle. But they showed pretty dramatically that they could rescue some of the effects of Rubinstein-Taybi even after birth. And that's kind of where I got drawn into this. I wanted to study Rubinstein-Taybi originally, but I couldn't get funding for it. So I kind of moved on to Kabuki syndrome. We've done a lot of work in Kabuki syndrome, and just to kind of remind you what we've done there, we established a mouse model called the beta geo model. And we started out by showing that the mice had many of the features that we've seen in patients with Kabuki syndrome. So low body weight, we could show that. The wild type are in blue, the Kabukis are in yellow. We could show that the immune deficiency that we see in patients with Kabuki, we see that in the mice too. And we were able to show that some of the craniofacial kind of things that we see in Kabuki, we could also see in these mice. So, you know, and the reason why I'm telling you is I think we're going to have to do the same with the Wiedemann-Steiner mouse model. We need to first establish that, look, we have a model that recaps what we see in the patients, and then we can kind of move on from there. From doing this, we were able to find some novel insights into the disease. And just to make it short, you know, one of the things that we found is that there seemed to be a defect of adult neurogenesis. So there's fewer, if you look at the green cells here, in the Kabuki mouse model, there's fewer of these green cells. So fewer cells are being made in the brain.
Hans, may I just ask a question at this point?
Of course.
So thanks, if it's okay if I jump in. Can you just describe what this marker DCX actually is? I'm not familiar with it, in fact.
Yeah, of course. The marker is called double cortin, and basically what it does, it's kind of an intermediate marker. It's not the first marker that neurons turn on as they go through the steps of adult neurogenesis, but it's kind of in the middle. And we've consistently seen that this marker, and not only this marker, but also the nice thing about the marker is you don't have to manipulate the mice in any way. The problem with the marker is it's dependent on this one protein, right? Double cortin. We're just measuring the amount of that. So we've replicated this by doing something called EDU injections, where we inject the mice with EDU. EDU incorporates into DNA, and then we get very similar distribution as this. The number of neurons that is being made in this area is always less in the Kabukis compared to the wild type. So, and we further were able to show, I hope that answers your question.
Yes, thanks very much. Yes, absolutely. Thank you very much.
Of course. So the other thing we saw in these mice is that the layer itself seemed thinner in the Kabukis compared to the wild type. And in addition, the mice had hippocampal memory defects. And it's very gratifying for Kabuki syndrome. People have now gone to patients and shown that in the patients, the dentate gyrus is smaller, and the patients also have these hippocampal memory defects. So some of these things that were novel in the mouse have now been replicated in patients, which is very, very nice. So this was kind of the things that we found that were novel. And then this allowed us to go then to the mice and do a strategy that was similar as the Rubinstein-Taybi strategy, which is just provide them with a powerful HDAC inhibitor, something called AR42. And here I'm just showing you sections from mice that are wild type on top, and then the beta geo, the Kabuki, on bottom. And you can see we see a big difference both in H3K4 methylation between the wild type and the mutant. And we also see a defect of these double cordon positive cells. They kind of show up as yellow-green here, and you see there's less of them here. And then as we increase the amount of the drug, the two genotypes got to be more similar. And this is a different cohort, but it shows the same response. As we gave more drug, the Kabukis in yellow, they caught up to the wild types for this marker. So seem to be making more neurons, or maybe moving neurons through stages. That could be another explanation. So then we followed this up with behavioral testing, and we had found that the Kabukis did worse, the Kabuki here in yellow compared to the wild type in blue. This is a task called the Morris water maze. So the mice are trained to find an invisible platform, and they use visual cues. We do this for a full week. And when we do this, we find that the Kabukis do about half as good as the wild types. However, when we put them on drug, they both did a little bit better, but also the Kabukis now were no worse than the wild type. So we felt like they were catching up with this. So again, AR42 is a very, very kind of big gun. So we thought of this as proof of principle studies, that this could show that we could manipulate Kabuki kind of in the postnatal life. So this is the summary for this introduction. Mouse model Kabuki syndrome shows impaired neurogenesis in the granule cell layer of the dentate gyrus. And these defects could be reversed using drugs that target the epigenetic machinery. And that suggests that aspects of the intellectual disability in Kabuki syndrome, and maybe some of these related disorders, could be treatable. So what are the caveats? The nice thing about having done a full study is that you can kind of learn where our limitations were. And these have been the complaints we've gotten with some of our work. Our Kabuki mouse is too artificial because it has this beta geo cassette. So, and it doesn't have a patient-specific mutation. That's a valid complaint. Another complaint that we've gotten is that the HDAC inhibitors have many actions, and maybe the treatment effect is non-specific and it doesn't work in the way that we think. That could very well be. We don't know if the treatment works in the long term. And I think by hindsight, if I was going to do it all over again, I would do a genetic rescue strategy just to kind of and try to use something that was close to a patient modification. I think that could just prove once and for all that you had postnatal malleability. And then many different people would, I think, jump on the bandwagon and develop, take their favorite therapy to this model. So that's kind of what I've learned from what we've done so far. So I wanted to take a little bit of time to talk about the biggest breakthroughs of the year. Obviously, I haven't been to the big meeting now, but these have kind of been creeping up as things that are worth to know about and have value for our work. So the first is CRISPR-Cas9, and you all know this. I spoke a little bit about this last year, and I'm going to just give you a couple slides about it, just a reminder. There's something called single-cell RNA-seq, which I think is very helpful. There's a new technique called cut-and-run technique. It's a new method to evaluate chromatin modifications. And then particularly for Wiedemann-Steiner, there came a paper where a group theorized that these two factors, KMT2A and KDM5C, were counteracting one another. And I'm just going to tell you a little bit about that because I think it's potentially relevant to Wiedemann-Steiner. So the first is, what is this CRISPR-Cas9 system? And so this is this amazing system that's been isolated from bacteria. It's kind of like an adaptive bacterial immune system. Bacteria and viruses have been fighting it out for millions of years or a very long period of time. And bacteria have developed this system where they take some of the nucleic acids from the virus infections and they put them in this array. And then whenever you get infected with that virus again, this sequence makes something called a cRNA, and then it forms a complex with something called a Cas protein, and it can cut the genome of the virus. And that's not so important, but some people figured out that this would be a really nice way to manipulate DNA. So, and now this system can be used to change any base in the genome, and it's going to have great ramifications for all genetic diseases, I think, not only for treatment, but also as you model things. And that's exactly what we've used with Jackson Labs, and I'll tell you about that in a little bit. But it rapidly changes our ability to make mouse models, cellular models, and treatments. And I think it's going to be a basis for a second wave of biotech revolution. This was so important that it made it to Time magazine in 2016. And this is a figure from a recent journal paper just talking about how CRISPR-Cas9 could be useful, making animal models, understanding genetic variation, making materials, food, fuel, and then they call something gene surgery, which is, I think, gene therapy or things like that. And this actually got the Nobel Prize in Chemistry this year, 2020. So I think people already see, most of the time you wait 20, 30 years for a breakthrough to get a Nobel Prize. But here, this is like a 10-year discovery or something like that. So people have really figured out how impressive and important this system is. The other thing that I wanted to tell you a little bit about is something called single-cell RNA sequencing. And we've used this. This is a way to study RNA. So RNA is what makes the proteins. And it allows you to study this in single cells, which is just amazing. Before, you had to have like thousands and millions of cells to study them. But now you can start looking at how cells behave as you differentiate them. And that's what I'm showing you here. This is from an iPS cell, an induced pluripotent stem cell, was another Nobel Prize not so long ago. But those are cells that you could take a skin cell and make it into a cell that can be differentiated into any cell. And here we've taken those cells and we're differentiating them into neurons, and we can track them during the process to see what gene expression is doing. And if we cluster the gene expression, what we see is the Kabuki line behaves very differently than the control lines. You can see this is mainly in the immature cells, more so than in the mature cells. And it doesn't seem to matter where they are in the cell cycle, but there's a big difference here, less of a difference here. And one of the insights that came from this is you can figure out where a cell is in this differentiation trajectory. And you can count how many for each genotype are at a highly differentiated stage or an early differentiated stage, early versus late. And when we did that in Kabuki, we found that the cells seem to run through differentiation faster than the control cells. So this is kind of giving you a different metric. It's giving you something about how rapidly the cells move through some process, which is something we couldn't do before. So I think this is a big breakthrough. And last year and this year, I think there's a lot of people using this assay to learn new things about new cell types and so forth. The third breakthrough I wanted to mention is something called cut and run, which I think is very relevant to these histone disorders. So cut and run is basically a method that allows you to characterize how much histone modifications there is at certain locations in the genome. And there used to be another method called ChIP-seq that everyone used, but the breakthrough here is that they can use much fewer cells for this method than before. So we can start doing this assay on cells directly from the brain or directly from animals. And I think that might be a way to figure out exactly how, Wiedemann-Steiner and Kabuki and these related disorders, what the underlying problem really is. So I'm pretty excited about this. The method came out in 2018, but there's just been an explosion this year in people using it, and it looks really, really good. So I wanted to mention that. The fourth Wiedemann-Steiner relevant finding was this paper that came out now 2020. It's from Shigeki Iwasa's group, and the title, like always, is a little bit cryptic. Mutually suppressive roles of KMT2A and KDM5C in behavior, neuronal structure, and histone H3K4 methylation. But what these guys did is that they had this hypothesis that KMT2A was competing with KDM5C, which is it's a demethylase, like an eraser that likes to remove this mark here, and KMT2A likes to place it. And what they did is they took mouse models that had a mutation in KMT2A and in KDM5C, and they bred them together. And they showed that the mouse that had both mutations did better than either mouse with a mutation. So it's basically a proof of principle that suggests that KDM5C would be a possible treatment target for Wiedemann-Steiner, that by inhibiting this enzyme, you might be able to treat some of the aspects of Wiedemann-Steiner. The only caveat that you have to remember is that this is treated from conception. So these mice either have both changes from birth or they don't. It's basically showing that, look, this could be treated if we could get in treatment at conception. It doesn't really prove that something could be treated later, but it suggests that this could be a valuable target for therapeutic development. So I thought this was exciting. I didn't believe everything they said in this paper, but I still thought overall it was pretty exciting and worth knowing about. I think the most important question still is, can Wiedemann-Steiner be treated after birth? And that's kind of what I'm going to focus on in the progress report, just to kind of give you a little bit of information about how we envisioned this project answering this question. And this is, I say here, taking advantage of prior experience. I think if I was starting over with the Kabuki stuff, this is the first thing I would do. And I have an old grant where I suggested this. I could never get the money to to get the mouse, but this was my early plan for Kabuki too. But it's a trick. It's a genetic trick. So we put in a stop mutation, which is what we see in most patients with Wiedemann-Steiner. And we use CRISPR-Cas to put in that stop into a mouse. But we also put these sites that are called LOX-P sites on either side. And the LOX-P sites are like these trick sites where you can use something called Cree to cut them out. So if we can give these mice Cree, and there are really good ways of doing that, we can get rid of the stop and we can ask, does the Wiedemann-Steiner mouse become a wild type mouse? Does it matter that it had this mutation during development? Can we rescue part of the phenotype or all of the phenotype by this? And this is always quite challenging because you not only need to put in this mutation, you also need to put in these special flanks. So it's pretty pricey, but I think it will answer this very, very important question. And for the proposal, and I'm amazingly thankful for your funding, we had been optimistic that we could start January 1st and then hopefully have mice ready at the end of 2020, so in December of 2020. And then we had proposed to start out by phenotyping the model, which is what I told you about Kabuki. We have to show that the mice have symptoms that are similar to patients with Wiedemann-Steiner or individuals with Wiedemann-Steiner. And then we could start asking, can we manipulate this locus? Does it work like we think it works? can we get rid of it with the special drug? And then the final, which is the most important, can we rescue disease aspects by removing the mutation after birth? You know, there were some things that have delayed us a bit, and this is probably the biggest culprit, the coronavirus, because it has really impeded all kind of international collaboration and transport and things like that, because different centers have been shut down at different times, and it kind of delays all the progress. But we've still made some progress, and I just wanted to walk you through the progress. The award was announced, I think, in March 2020. We reached out to Amir Subiri as soon as that was done. It took a while for us to establish contact. It was a really good idea to contact him because of your interaction with him, he had already been kind of a little bit interested in Wiedemann-Steiner, and he's actually made another mouse model there, which is available to us and soon to other people, which could be a backup model. Let's say our model doesn't pan out, then we could do our studies with that model. We did the due diligence to get multiple offers, but because of him being kind of committed to Wiedemann-Steiner, we decided to go with JAX and have the possibility of that backup model. That's a really nice safety mechanism. If we can't get this to work, we have this other model that we could phenotype and look at with drugs and so forth. We got the funding, I think, in-house in May 2020. It took my institution a little bit of time to go through all the paperwork. And I was able to get a student to help with some of the design things pretty quickly, and we were able to order these mice in June 2020. And this was the timeline that Amir and his group gave us. Order went in June. In August, they injected some of these embryos, and then the hope is that November we would have mice based on this current timeline. I reached out to Amir before this meeting, and it turns out that there are 6 mice that have already been created. They're all born the 9th of September, so they're pretty close to be able to breed. And so what needs to happen now is they need to make sure that the mouse transmits the mutation. It looks like the mutation that we wanted was created for all 6, but now we have to prove that the mice make offspring that can survive, and that you know, we can propagate the line. So I should know that in the next month or so. So the summary for this is just that, you know, we'll know in the next month or 2 whether this strategy worked. We're only going to pay if we get viable mice. That's the deal that we have with JAX. So we have this backup mouse if that doesn't work out. I'm hoping that we can transport the mice to Iceland in January. I don't know how this coronavirus thing is going to affect things in the next couple of months, but a lot of things are back to business at this point, even though things were shut down earlier. But now there's a 3rd wave in the US and so forth. So I don't know if this is going to be a major source of delay, but there has been an ability to transport animals so far. And then once we get the mice to Iceland, we'll expand the colony. It will take us 1 to 2 months, and we'll get started on experiments. So the bottom line is we're delayed because of the COVID and various things, but we're not horribly delayed. I think we're a couple, maybe 2 to 3 months behind, but hopefully we can kind of catch up, you know, as soon as we get the mice. So these are just some of the people that participated, and then this is my website and my email. So if you have any, if anyone that's listening has any questions or comments or want something clarified, they can just shoot me an email. So that's all I have to say. I'm happy to take any questions.
Thank you very much. Can you hear me? If I may, I don't know if there's any burning questions. I would probably just get started. Yeah, I scribbled a few things down, so I wanted to. Can you just one point that springs up in slide 28, which you have here, slide 28, your last slide, where you described the viability of the mice. So in terms of the term, when we use the term viable, we mean just simply, they don't guarantee functionality of the allele, do they? They just mean that they basically, they assuming the mutation, I mean, because, you know, bad things can happen, but so they guarantee that if you have live mice where this allele is carried, that's how they deem viable mice, not if you cross it to a Cree and the LOXP doesn't flick out, if it doesn't recombine. That's not, do I understand that correctly?
Yeah, you're absolutely correct. Yeah. You can still get the cat in the box, you know, or the cat in the sack or whatever. The nice thing is that Amir, because he has a little bit of interest here, he's been willing to take over the mice at this stage and is going to do some preliminary tests with, you know, see if the LOXP works and so forth. But again, you know, they promised nothing really. They just say, you know, when I spoke to him, and I had many meetings with him, because in the beginning he said this can't be done, and I showed him some papers where people had done this, and then he said, well, okay, it can be done, and if we're going to do it, these are my thoughts. And he was quite helpful in helping us, you know, design this. But again, there's a couple of different caveats that I foresee, and this is maybe not sounding very optimistic on my behalf, but the first thing is it doesn't look like a Wiedemann-Steiner for some reason, and it doesn't have any of the phenotypes, and you know, that's one possibility. I don't think that's going to happen because of his other mouse. He already has a mouse that has a little bit decreased viability, but it's a similar mouse as the full mouse here. So it's a loss of function allele. So I think these mice are going to have.
Oh, has anyone else lost Hans for the moment?
Yes.
Okay, I guess he'll reconnect in a moment. I wonder if he's lost us too.
I think Hans is frozen still for me.
Yeah.
We could overcome that that difficulty with the Cree, because we could give the Cree early on, and that could be another way of kind of measure. It's not exactly what we plan to do, but that's one way to kind of salvage the line and boost the numbers of the line. So
I can ask another question then at this point. So what we have here is essentially a knock-in for this LOXP flank stop cassette between, I guess, the promoter and exon 1, as I understand the strategy of the mouse generation. Is that correct? So in principle, all of these founders should potentially be now haploinsufficient, right? For as they stand.
Yeah. Exactly. The founder mouse is a full, you know, is basically a haploinsufficient. Absolutely.
And so do we have any information to the sort of the body weight of these founders? I mean, we could already get a hint if they are progressing normally, which would maybe indicate that they're not completely recapitulating, you know, haploinsufficiency at this point, or if they're maybe. Because I mean, what another thing that I've noticed from the literature, because there've been a couple of people who've made KMT2A knockout mice in the past, and the phenotypes seem to be quite variable, no? For unexplained reasons. You have some which have rather strong phenotypes, and some which have rather less severe phenotypes, if I remember correctly at this time. And I don't think anybody understands quite why that is. So even if these don't look like they have a very strong phenotype at this stage, it could still be very, very useful for crossing back to another haploinsufficient strain where you could restore the normal function, I would imagine. Right?
Yeah, I think he doesn't want to phenotype these because they could be mosaic, the way the CRISPR is basically injected into the embryo. But the next generation, that's his plan, to look whether they have some of the same phenotypes, growth, other problems.
Okay.
And, you know,
I see.
whatever he's willing to do, I think would be great, because he has a lot of tools in house. But you know, it didn't sound like he would do a lot. He said he'd take them over, he'd breed them, you know, to next step, and he'd do some basic testing of whether he thinks that the LOXP sites are working, and whether the mice have some basic phenotypes that we've seen before. The litter size, so he has this other model that we can get from him, and there it's pretty striking that they have Wiedemann-Steiner phenotypes. They, you know, there's a decreased litter size, but the mice are smaller, and there's been some vague neurological problems in those mice. So you know, again, that backup mouse I think could be useful and hasn't been phenotyped yet. So and it's very similar to most of the mutations we see in the patients, which are these loss of function mutations.
Okay, and these are the mice where he's basically created a substitution, correct? Where he's just taken, I think it's somewhere in the, yes, I have this generation of the C1186Y mutation as a clinical allele in the murine KMT2A gene. So I think what he's done is he's essentially made a substitution which results, I believe, in a truncated KMT2A. Is that correct? And that looks good, you say, because I was in contact with him about half a year ago about this, and when he told us that he'd actually begun all these projects, and or that there was already made a lot of progress in these projects. So I'm glad to hear that really looks good. I mean, if these mice are viable and you can breed them, that's great. That's really good news.
I think that, and we'll see how expensive it is to get those mice too, because maybe when we ship, it may be worthwhile to just get this model. We're going to need to order a tamoxifen model too, so, or something to give the Cree. So you know, we may take all three over, but that, I think it's a really nice backup. And Jax is going to share that model not just with us but with the community. So I think that will be super valuable.
And he's also making a flox, is he not? KMT2A flox, I think. I don't know if you're aware of that. I think he's trying to make also a conditional KMT2A.
Yeah, I think there has been some success with that as well.
Okay.
I'm not completely sure where that stands, but
absolutely. I think it's a much longer process, I think, to make a floxed allele. I think it's a bit more tricky. If I understand correctly,
it's tricky, and also, you know, but what the flox allows you to do is you can knock out now KMT2A in any given cell type to try to ask the questions, what are the most critical cells? So it will be a useful model. And my hope is that I can use your funding to get a bigger grant. You know, if we can have some preliminary data, we might be able to get, you know, an additional grant to kind of build upon that, which is, I think everyone should always consider. The funding is great, but it's not a lot of money for an ambitious mouse project, you know, so.
Yeah. Maybe I can ask, so I guess you have, you know, plans. I mean, in the event that, let's say, the allele wasn't functional for some reason, is it then, and we were to get this, but I mean, hang on a minute, we've already kind of, so if we get germline transmission now, we've guaranteed viable offspring of the allele.
That's correct.
Which I think is probably likely based on what we see here. I mean, unless there's something that we don't know. I don't know how. I mean, these mice are now what? They must be 6 weeks old now.
Yeah, and
yeah,
the fact,
I think we.
Usually, when this step fails, you get 1 or 2 founders. And then the founders, because I think these are a couple different injections that these came from. So
Okay.
I'd be shocked if we don't get viable animals. You know,
I think, yeah. Mm-hmm.
It would be a great surprise to me. So I think
Mm-hmm.
we're probably, whether the model works exactly as we planned, I don't know about that, but I think, you know, the fact that we have founders and this many founders, I think we probably have viable animals.
Yeah, I think an easy way to work out if the LOXP, well, one of the easy ways to work out if these alleles are working is to just simply intercross. Once you have offspring of one of the founders, if you cross the males and the females together, you should end up with no homozygous animals, correct? Because it should be, you know, a full knockout is then embryonic lethal.
Yeah.
That could be one way to address that, right? Just to simply, you intercross from the founders, and then the one in which you have basically no homozygous mice, you would know that was then truly blocking the KMT2A function, right? That would sort of guarantee that your LOXP sites are working. Of course, I mean, not just, you can always do these PCRs and get this recombination, and you can work that out, but it doesn't mean you're blocking the expression of the gene, sadly. There are ways that could be taking place. So maybe that's, I mean, but you know, they will think of that. I think we just need a bit of patience now to work out which one of these founder lines will be efficient and will work. So yeah, I think by the end of the year we'll have much more information.
I think
yeah.
You're right. I think
anyway, they will have thought of that at JAX to intercross. They will have thought of that for sure. Okay. Yeah. Okay.